all, today we will be doing Module 34, Investigation of Foodborne Illness Outbreaks. This particular module will be covered under various subheads such as why there is a need for investigation, the objectives of investigation, the personnel required, what are the other requirements like materials and equipments. We are also going to talk about the investigation procedure, the major steps. We are also going to see how investigation takes place, and the focus is on the environment, food and the laboratory investigation. We are also going to discuss the preventive and the control strategies. And in the last, the entire module will be concluded or summarized. Now, before we start this module, we should know what is a foodborne illness outbreak. The definition should be known so that we can build the entire module around this particular outbreak. It is an incident in which two or more people experience a similar sickness after ingestion of a common food. This food can be a solid or a liquid. There is definitely a need for investigation. Why do we need to investigate? First of all, to avoid or to prevent transmission of ongoing disease as well as related outbreaks in the future. We do not want the outbreaks to take place. Therefore, we need to investigate. We also need to see why this has occurred. It has resulted from the consumption of which fruit, vegetable, dairy, meat, fish or seafood. We will also focus on liquid foods. They can be milk, water or any beverage. The symptoms of most of the illness, they are dependent upon the etiological agent. When we talk about the investigation of a foodborne illness, we should have certain objectives or goals in mind. The first is we need to recognize the possible factors which have played a significant role in the contamination, survival or proliferation and spreading of this particular agent. Then we need to also talk about strengthening of food protection or food safety guidelines as well as to prevent similar outbreaks in future. We need to acquire the data how this particular agent has led to the contamination, and we also need to record and document with regards to the assessment of the causative agent and the risks involved. We need to encourage the research studies which will assist us to prevent related outbreaks in the near future. When we are talking about the objectives, we also need to control and prevent any enduring outbreaks. We need to recognize and exclude the implicated or the suspected foods. We also should have methods of precise detections and identification of possible risk factors or threats. These threats can be associated to the host or the pathogens or the environment. We need to look in a very holistic way onto the host, onto the pathogen, from where it is coming from the environment. When we want to investigate a foodborne illness outbreak, we should have a team which should be comprising of 3 groups. The first and the foremost is the in charge officer. Then he should also have a group of members who are going out in the field and collecting information. Then the other group which is going to work in the laboratory. Each personnel which is involved in the specific category should be assigned a specific work. When we are doing the investigation of a foodborne illness outbreak, we need lots of backup in terms of infrastructure. So when we talk about the infrastructure, we should have infrastructure for picking up the information or the samples from the premises where this has happened. We will be needing sterile containers, sampling devices. We will also require sterile swabs In diluents, we'll also require disinfectants like spirit or alcohol. We might need an alcohol lamp also, a thermometer, we might need papers, we might need tapes, chest for bringing the samples back to the lab, and forms for data recording. Apart from these basic requirements, we should have a lab which is well equipped. When we talk about a lab, it should have ample supplies of glassware, plasticware. We should have lots of culture media. We can detect various types of pathogens, indicators of safety, indicators of quality. We should also have equipments like autoclave, laminar airflow chamber, microscope, hot air oven, weighing balances. When we are analyzing an outbreak due to a chemical agent, then we might need sophisticated equipment like HPLC, GLC also. When we talk about these illnesses, we are dividing this investigation process into two parts. The first thing is the field investigation, which will comprise of epidemiological investigations, environmental and food investigations, and then all the investigation will start taking place in the laboratory. So when we talk about a foodborne outbreak, we should be knowing the process. The process is complicated, but it is a very step-by-step process which is thoroughly interconnected. First of all, there will be a reporting of the food illness outbreak. It can be a hospital case or it can be a phone complaint coming in. Then there are chances foodborne illness can be a single or it can be a case where multiple people are involved. Usually a 74-hour meal report is taken, notified, and report to outbreak report is done to the investigating team members. At times we also see the weather which was around, because at times it also plays a culprit in causing a foodborne outbreak. This is usually in the month of summers. After this, the field investigation starts and the field team members, they visit the particular premises, they interview and they prepare a case history from the subjects. Subjects means patients who have fallen sick or ill, and also other people who have not fallen ill after the consumption of that particular food. Then there is collection of food samples and they are sent to the lab. All the specimens are collected. Even the health of the food handler or the person who was distributing food is taken into consideration. Once the samples they reach the laboratory, then the laboratory investigation starts, and it can comprise of microbial and chemical analysis. We are responsible here to identify the etiological agents which have led to the outbreak of this foodborne illness. After the laboratory investigation is done, there is a development of a case and we identify a particular causative agent. After that, there is a formulation of a hypothesis. All the details are being recorded, and if there is a need of further investigation, it takes place, and interview of additional cases also takes place. Additional cases means the other people who have been identified during the hypothesis process. After this, the collection of data takes place. There are calculations and analysis of that data. Once the calculations are through, the hypothesis is tested and a corrective measure is suggested to control the outbreak. After that, a report is written and submitted to the authority for further action, and later on this particular report can also be used for outbreak of such kind of diseases in near future. Now we will be talking about major steps involved in the investigation of foodborne illnesses, how they are recorded. First of all, there is a role of the local health department. It needs to be notified of the illness by phone or by the hospital where the patients have gone. Now there can be two types of cases happening. In one case, a single person is involved, or other case we can have number of people who have fallen sick after consuming this food. The time period of 72 hours is very important, and all the complaints are reviewed, the data is collected, and further action is being carried out. After 72 hours are over, we decide to document the reason for action, we decide whether to investigate further or not, or we need to prepare an investigation report and we should send it to the disease reportable section. Now when we talk about a disease and we feel that further action has to be taken, then we decide to obtain a case history, that is usually done by a survey. Then we conduct inspection of the facility from where the food was procured or cooked. Then we need to collect the food water samples. We need to check the health status of food handlers and also obtain the clinical specimens. All these steps are very critical, and they help us to investigate the foodborne illness in detail and in depth. The next step is to develop a case definition and identify the cases. Once this is done, then we make a epidemiological association, that is time, place, person. Once this is done, a hypothesis is formulated and we expand the investigation process. We find and interview additional cases and persons who can be at risk. After that, the data is collected, the calculations are made, and we go in for the further analysis of the data. We are again testing the hypothesis in depth. We are taking corrective actions. We are writing a report, putting information, and this report is being submitted to the department, especially to the reportable disease section. And this particular data can also be used further to prevent such outbreaks. When we are doing the investigation of a foodborne illness, environment, food, and the sanitary investigation are also very important. These are carried out to identify the source, the mode of transmission, and extent of food contamination. We are also evaluating the possibility that the pathogen or the chemical, how it survived. We are also trying to focus on the methodologies and processes designed to destroy or inactivate them and to diminish their concentrations. We are also trying to find out the potential of pathogenic microorganisms to grow or survive during processing as well as storage of foods. Last but not the least, we are also trying to identify and implement the remedial methods. Now we come to a very important part of foodborne illness outbreak investigation that occurs in the lab. This is the laboratory investigation, and this is very important because majority of foodborne illnesses are microbiological in origin. Therefore, there is a need for a well-equipped microbiology lab. When we talk about the microbiological examination, a simple test like gram stain can be used as a prime method for initial screening of microbial pathogens. When we talk about the methodology, it should be as per the standards and guidelines of the regulatory bodies. Most of the times in microbiological analysis, we are using the conventional microbiological techniques, which are very time consuming. Therefore, lots of work is being done for faster methods, which are advanced molecular biotechnological techniques, which are used for rapid identification of microbial species. One such technique is real-time PCR, which gives us a quick detection. Identification also helps us in enumeration of pathogenic microorganisms. Lots of work is being done in this field, and very soon we'll have cost-effective methods which will be able to help us in identification, to the subspecies level. So when we are talking about the laboratory investigations, these outbreaks, they can also be caused by chemically contaminated foods. Although they are very common, but they are, when we compare them to the microbiological incidences, they are slightly less. But when we are talking about an outbreak which is due to a chemical agent, our requirement is slightly different. We will be depending upon the primary laboratory testing for suspected chemical agents. There is a need for sensitive, reliable and reproducible results. We might need advanced technology like GLC, HPLC, ion exchange chromatography to identify the chemical agent which has led to this particular outbreak. When we are talking about the investigation, prevention is always better than cure. So we should be taking care of certain preventive and control strategies. These can be the proper sanitary practices during food manufacturing processes. We should subject the food to right and adequate heat treatment. We should keep an eye on the time and temperature. We need to avoid contamination from infected food, food handlers or carriers. We should be able to have a connection among a particular food and transmission of the suspected pathogen. Once it is established, then it is important to close down the food premises. We need to prohibit the sale of food and we need to remove the implicated foods from the market. We also need to modify the process of food preparation, which is very, very important so that future outbreaks, they do not take place. So when we are talking about the preventive and the control strategies, there should be appropriate recommendations and guidelines among the people regarding avoidance of chemically contaminated food, water, or they also need to understand that a simple step like boiling of microbiologically contaminated food or water can save them. A well-structured appraisal by the outbreak control team, that is OCT, is very, very important. The OCT needs to prepare an interim report by 2 to 4 weeks after the outbreak so that by the end of the inquiry there should be a final written report which will play a very important role in deciding how the future outbreaks can be tackled or stopped. So in the end of this module 34, it's very, very important to understand that prompt reporting of an outbreak to the health department is most important. The first task is to gather all the necessary information about the cause of an incidence, including the kind of food, the targeted age group, the time of food consumption, history, and other important variables such as the food handlers, the water source, the location of the premises. It is very, very essential to reject suspected foods to prevent further spread of illness or infection. Education of the public is very, very important because once they are aware, those practices which are leading to the disease outbreak, they can be curtailed and they can be stopped. When we are talking about the OCT team, they have a very big role to play because their report should be comprehensive, confidential, and it should be circulated to the right authorities and individuals so that in future these kind of foodborne illness outbreaks, they do not take place. Thank you.